Welcome to the deep dive. Today, we're really getting into the weeds, tackling something absolutely fundamental: how our bodies, right down at the cellular level, generate the energy that keeps us all going.
Exactly. We're talking about mitochondrial respiration and oxidative phosphorylation. It's basically the power plant inside almost every one of our cells.
The power plant, I like that. And understanding it is crucial, isn't it? It's directly linked to our energy, our health.
Absolutely. And when it goes wrong, maybe through inherited defects or, the effects of drugs or poisons, the consequences can be really devastating. So getting a handle on this machinery gives you a real insight into, well, how life fundamentally works.
Right. So our mission today, for you listening, is to achieve a really solid, almost professional level understanding of this incredibly intricate process. We want you to walk away feeling like you get it.
It's a complex dance, but we can break it down.
Okay, so here's the plan. We'll start with the mitochondrial structure, the stage for all this. Then we'll trace the path of electrons through the transport chain.
Uncover how ATP is actually made.
Look at how it's all regulated, what can block it, the inhibitors, and finally how molecules get moved around, in and out.
We've synthesized a lot for this core biochemistry, specific examples. It should be comprehensive.
Great. So let's start with the stage itself, the mitochondrion. What's the basic layout?
Okay, so picture this. It's got a double membrane, an outer one and an inner one.
Two layers.
Right. The outer membrane is fairly permeable, lets smaller stuff through pretty easily. But the inner membrane, that's the really critical barrier.
More selective.
Highly selective. It controls exactly what gets into the very center, the matrix.
Yeah.
And it's not just a smooth bag, it's folded extensively. These folds are called cristae.
Yes, the cristae. Why the folds?
It's all about surface area. Dramatically increases the space available for all the protein machinery. We're going to talk about the electron transport chain, ATP synthase. They're embedded right in that inner membrane. This compartmentalization, this separation by the membranes is absolutely key for setting up the energy gradient.
Okay, so like setting up different zones in a factory. Where are the specific tools, the enzymes, located within these zones?
Good analogy. Yeah, different parts have different jobs. On the outer membrane, you find enzymes like acetyl-CoA synthetase, that's for starting fatty acid breakdown, and glycerol phosphatidyltransferase for lipid stuff.
Okay.
Then in the space between the two membranes, the intermembrane space, you've got things like adenyl kinase and creatine kinase. We'll maybe touch on that later with the shuttle.
Right.
But as I said, the intermembrane is really the main event. That's where the respiratory chain complexes through the FOVR, the ATP synthase machine itself, and loads of transporters. Oh, and a special lipid called cardiolipin, really important for keeping those protein complexes stable and working properly.
Got it. Intermembrane is action central. So within this factory, what's the core process happening in the innermost part, the matrix?
Right, the matrix. That's where the main fuel burning happens. Oxidation of substrates, carbs, fats, proteins broken down. This oxidation generates high-energy electron carriers, mostly NADH and FADH2.
These are like the energy delivery trucks, carrying electrons.
Exactly. Loaded trucks full of high-energy electrons arriving from fuel breakdown processes like the citric acid cycle, which also happens in the matrix.
Okay, trucks loaded, arrived at the inner membrane area. What happens to the cargo of those electrons?
This is where the respiratory chain or electron transport chain comes in. It's a series of 4 big protein complexes, complex 1, 2, 3, and 4, embedded right there in the inner membrane.
The ones you mentioned being on the inner membrane.
Precisely. Their job is to take those high-energy electrons from NADH and FADH2 and pass them along like a baton in a relay race through a series of controlled redox reactions.
And where do the electrons end up?
The final electron acceptor is molecular oxygen, O2. It gets reduced to water. And this is crucial as the electrons move through some of these complexes, specifically I, 3, and 4, something else is happening.
The proton pumping you hinted at.
Yes. These complexes use the energy released as the electrons drop in energy level to actively pump protons, hydrogen ions, H+ from the matrix out across the inner membrane into that inner membrane space.
Pushing protons outwards. Why? What does that achieve?
It achieves the creation of a gradient. You end up with a higher concentration of protons in the intermembrane space than in the matrix. It's also an electrical gradient because you're moving positive charge out, so the outside becomes more positive relative to the inside.
So it's like building up pressure, storing energy.
Exactly like that, like water building up behind a dam. This combined gradient chemical concentration difference plus electrical potential difference is called the proton motive force. It's stored potential energy.
And that's the energy that's ultimately used to make ATP.
Precisely. That stored energy in the proton gradient is what drives the next final step, oxidative phosphorylation. The protons flow back down their gradient through another protein complex, ATP synthase, and that flow is harnessed to make ATP.
Okay, brilliant overview. From fuel oxidation to electron carriers, electron transport, proton pumping, gradient formation, and finally ATP synthesis using the gradient. Let's zoom in on the electron transport chain itself now, those four complexes.
Right, complexes I through 4 embedded in the inner membrane. The main path for electrons, starting from NADH, goes complex 1, then to a shuttle molecule called coenzyme Q.
Coenzyme Q, also called ubiquinone.
Yep, same thing, ubiquinone or CoQ. Then from CoQ to complex 3, then to another mobile carrier, cytochrome C, and finally to complex 4, which hands them off to oxygen. It's a very specific sequence.
And you mentioned FADH2 takes a slightly different route.
Correct. FADH2 comes from reactions like succinate oxidation in the citric acid cycle. Its electrons have slightly less energy, so they enter the chain via complex 2. Complex 2 also passes electrons to coenzyme Q. So CoQ collects electrons from both complex 1 and complex 2.
But complex 2 itself doesn't start the chain for NADH.
No, NADH goes to complex 1. FADH2 goes to complex 2. Think of complex 2 as an alternative on-ramp to the CoQ roundabout.
Okay, got it. And these mobile carriers, CoQ and cytochrome C, are crucial for linking the big complexes.
Absolutely essential. Coenzyme Q is small, lipid-soluble, so it moves within the membrane, shuttling electrons between I and 3 and 3 and 3. Cytochrome C is different. It's a small, water-soluble protein in the intermembrane space. It ferries electrons from complex 3 to complex 4.
Different locations, different properties, perfect for their specific shuttle jobs. Now, inside the complexes themselves, you mentioned other key components. Flavor proteins and iron-sulfur proteins. What do they do?
They're prosthetic groups, basically helpers bound within the protein complexes that actually handle the electron transfers. Flavor proteins use FMN or FAD. You find these in complex I, FMN, and complex 2, FAD. A key thing about flavoins is they can handle electrons 1 or 2 at a time, which is useful for connecting carriers like NADH, 2 electrons to 1 electron carriers down the line.
Flexible electron handling. And the iron-sulfur clusters.
Iron-sulfur proteins, or SES proteins. They're all about single electron transfers. They contain iron ions linked to sulfur atoms, either inorganic sulfide or cysteine residues in the protein. The iron cycles between Fe2+ and Fe3+ states to pass the electron along.
So they act like stepping stones for individual electrons.
Exactly. And there are different arrangements, 1Fa, 2Fe2s, 4Fe, 4s clusters found in complexes 4, 2, and 3. They have slightly different properties, fine-tuning the electron flow. Complex 3 even has a special Rieske iron-sulfur center, which is crucial for its unique function in the Q cycle.
Okay, packed with these specialized electron handlers. Let's walk through the complexes one by one, focusing on that coupling electron flow linked to proton pumping. Start with complex 1.
Complex 1, or NADHQ oxidoreductase. It's a giant L-shaped complex. Its job is to take 2 electrons from NADH in the matrix and transfer them to coenzyme Q within the membrane. And importantly, for every NADH it processes, it pumps 4 protons from the matrix out to the inner membrane space.
4 protons right off the bat from NADH. How does it manage that transfer and pumping?
It's intricate. NADH passes electrons first to FMN. Then they hop along a series of, I think, at least 7 different iron-sulfur clusters, each with a slightly different energy level, guiding them towards CoQ. CoQ picks up the 2 electrons and 2 protons from the matrix to become QH2. The exact mechanism linking electron movement to proton pumping is still debated, but it definitely involves conformational changes in the protein structure, like parts of the pump moving as electrons pass through.
Fascinating. 4 protons per NADH. Now, complex 2, you said it's different.
Right. Complex 2, or succinate Q reductase. It's the entry point for electrons from FADH2, which is generated when succinate is oxidized to fumarate in the citric acid cycle. In fact, the enzyme doing that, succinate dehydrogenase, is part of complex 2.
It's integrated.
It is. So FADH2 passes its electrons through 3 iron-sulfur centers within complex 2 and then onto coenzyme Q, reducing it to QH2. But here's the key difference. Complex 2 does not pump any protons.
No protons pumped at complex 2. That's a big deal for energy yield later, isn't it?
Absolutely. It means electrons entering via FADH2 contribute less to the proton gradient compared to those from NADH entering via complex 1.
Okay. And are there other ways electrons can get to CoQ bypassing complex 1?
Yes, there are. Electrons from breaking down glycerol 3-phosphate, important in some tissues, or from fatty acid oxidation via acyl-CoA dehydrogenase also feed into the CoQ pool. These use other F80 link enzymes, and like complex 2, these pathways don't pump protons at that initial entry step either.
So CoQ is like a central collection point for electrons from various sources.
Yeah.
Now, once QH2 is formed, loaded with electrons from complex 1 or 2 or these other paths, where does it go? Complex 3.
Correct. Complex 3, or Q-cytochrome C oxidoreductase, its job is to take electrons from QH2 and pass them to cytochrome C. Now remember, QH2 carries 2 electrons, but cytochrome C can only carry 1 at a time.
Ah, a mismatch. How does complex 3 handle that?
Through a really clever mechanism called the Q cycle. It's a bit complex, but it essentially allows complex 3 to handle the 2 electrons from QH2 sequentially, pass them to 2 separate cytochrome C molecules, and pump protons in the process.
Okay, walk us through the Q cycle. Sounds important.
Right. So first QH2 binds, it gets oxidized, its 2 protons are released into the inner membrane space, contributing to the gradient. 1 electron goes up via the Reskifié S center and cytochrome C1 to reduce a molecule of cytochrome C.
Okay, 1 electron transfer, 2 protons released. What about the second electron?
The second electron goes down through 2 different cytochromes within complex 3, cytochrome BL and BH, and partially reduces another CoQ molecule that's bound at a different site, forming a semiquinone radical, Q1. This picks up a proton from the matrix.
Still only halfway there, right? Need to reduce a second cytochrome C.
Exactly. So a second molecule of QH2 binds and does the same thing. Releases 2 protons out, sends 1 electron up to reduce a second cytochrome C. Its other electron goes down again, finds that semiquinone radical, Q, reduces it fully back to QH2, and this step picks up a second proton from the matrix.
Wow. Okay, let's sum that up. 2 QH2 molecules get oxidized.
Right, releasing a total of 4 protons to the intermembrane space. 1 CoQ molecule gets reduced back to QH2, consuming 2 protons from the matrix, and crucially, 2 molecules of cytochrome C get reduced.
So the net effect is transferring 2 electrons from QH2 to 2 cytochrome C molecules while effectively pumping 4 protons across the membrane. That's elegant.
It really is. It solves the 2 electron to 1 electron carrier problem and pumps protons efficiently.
Okay, so reduced cytochrome C is ready. It ferries its electron to the final player, complex 4.
Complex 4, cytochrome C oxidase. The end of the line for the electron transport chain. It takes those electrons one by one from cytochrome C, and it uses them to do something absolutely vital: reduce molecular oxygen O2 to water, H2O.
This is where the oxygen we breathe comes in.
This is it. This reaction consumes the electrons and protons to make water. It requires 4 electrons, which means 4 cytochrome C molecules donating 1 electron each, to reduce 1 molecule of O2 completely.
And does complex 4 also pump protons?
It does. As those 4 electrons pass through complex 4 to oxygen, it also pumps 4 protons from the matrix to the intermembrane space.
4 more protons. Okay.
Oh, yeah.
How does the electron flow work within complex 4?
It involves several metal centers. Electrons come from cytochrome C to a copper center called CuA, then to a heme group called heme, then to a special binuclear center made of heme 3 and another copper ion, Cub. This A3 Cub center is where oxygen binds and gets reduced.
It sounds like holding onto oxygen tightly during this process would be important to avoid releasing nasty intermediates.
Extremely important. Complex 4 holds the oxygen molecule very tightly at that binuclear center until it has received all 4 electrons. This minimizes the release of partially reduced and potentially very damaging oxygen species like superoxide radicals or peroxide. It's a crucial safety feature.
Makes sense. Okay, let's quickly recap the proton pumping tally per electron pair, like from one NADH. Complex I pumps.
4 protons.
Complex 2.
0.
Complex 3 via Q cycle.
4 protons.
Also pumps protons. For the 4 electrons needed to reduce 1 O2 molecule, it pumps 4 protons. So per electron pair, like from NADH, that's effectively 2 protons pumped by complex 4.
So for NADH going through I3 and 4, that's 4 plus 4 plus 2 equals 10 protons pumped per pair of electrons.
Roughly, yes. That's the typical number used in calculations. For FADH2 starting at complex 2, it's just 0 plus 4 plus 2 equals 6 protons pumped.
Okay, this proton pumping has built up that powerful proton motive force across the inner membrane. Now, how is that energy tapped? Enter ATP synthase.
Exactly. All that work building the gradient pays off here. The chemiosmotic theory, Peter Mitchell's brilliant idea, explains it. The protons naturally want to flow back down their concentration and electrical gradient from the intermembrane space back into the matrix.
Back across the inner membrane. Right. And the only way they're allowed back across efficiently is through a specific channel provided by the ATP synthase enzyme complex.
And ATP synthase isn't just a channel, it's a machine, right? A turbine.
An amazing molecular machine. A turbine is a great analogy. It harnesses the energy of that proton flow, the proton motive force,
Yeah.
to drive the synthesis of ATP from ADP, an inorganic phosphate.
Pi. Tell us about its structure. You mentioned two main parts.
Yes, F1 and S0. The F0 part, that's O for oligomycin sensitive, not zero, is embedded in the inner membrane. It forms the proton channel. It's made up mostly of a ring of identical C subunits plus some other connecting subunits.
The part sitting in the membrane. And F1.
F1 sticks out into the mitochondrial matrix. It looks kind of like a lollipop head. This is where the actual ATP synthesis happens. It's made of several different subunits, notably 3 alpha and 3 beta subunits arranged alternately, like segments of an orange. The beta subunits contain the catalytic sites for making ATP.
And how are F0 and F1 connected? How does proton flow in F0
Yeah.
make ATP in F1?
They're connected by a central stalk made of gamma and epsilon subunits. This stalk runs from the rotating C ring in S0 right up into the center of the F1 head. As protons flow through the F0 channel, they bind to sites on the C subunits, causing the whole C ring and the attached central stalk to rotate, like a water wheel turning.
The proton flow literally spins part of the enzyme.
It literally does. And as that central gamma stalk rotates inside the stationary F1 head, which is held in place by another peripheral stalk, it bumps against the inside faces of the 3 beta subunits, forcing them to change shape.
Change shape. How does that make ATP?
It's called the binding change mechanism. Each beta subunit cycles through 3 conformations: open O, loose L, and tight T. In the O state, it can bind ADP and pi or release ATP. In the L state, ADP and pi are loosely bound. As the gamma stalk rotates, it forces a change to the T state. In the T state, ADP and pi are bound so tightly that they spontaneously form ATP. Then another rotation forces it back to the O state, releasing the newly made ATP.
So the rotation drives these conformational changes. And one of those changes essentially squeezes ADP and pi together.
That's a good way to think about it. The energy from the proton flow is transduced through mechanical rotation into the chemical energy of ATP's phosphate bond. Each full rotation of the gamma stalk causes each of the 3 beta subunits to cycle through OLT, producing and releasing 3 ATP molecules in total per revolution.
Incredible. A nanoscale rotary motor. Now this brings us to PO ratios. You hear numbers like 2.5 ATP per NADH. How does that relate to the protons pumped in this rotation?
Right. The PO ratio is ATP made per oxygen atom reduced, or per electron pair. We said NADH oxidation pumps about 10 protons, and making and releasing 1 ATP via synthase takes about, well, the number varies slightly depending on the number of C subunits in the ring, but let's say roughly 4 protons need to flow back through ATP synthase, 3 for synthesis, 1 potentially for phosphate transport. So 10 protons pumped per NADH. 4 protons per ATP gives you roughly 2.5 ATP per NADH.
And for FADH2?
FADH2 only leads to about 6 protons being pumped, bypassing complex I. So 6 protons pumped, 4 protons per ATP gives roughly 1.5 ATP per FADH2. But these are theoretical maximums, mind you. Real-world yields might be slightly lower due to things like proton leaks across the membrane.
Okay, that clarifies the PO ratios. Now the cell isn't always working flat out. How is this whole process controlled? How does ATP production match demand?
That's respiratory control. It's fundamental. Under normal conditions, electron transport and ATP synthesis are tightly coupled. Basically, electrons don't flow down the chain unless protons can flow back through ATP synthase. And protons only flow back readily if ATP synthase is actively making ATP.
And what determines if ATP synthase is active?
Primarily, the availability of its substrates, ADP and pi, especially ADP.
Right.
When the cell uses ATP for work, it generates ADP. This ADP signals the need for more ATP. It enters the mitochondria, binds to ATP synthase, and essentially says, go. Protons flow, ATP is made, electrons flow, oxygen is consumed.
So ADP levels act like the accelerator pedal.
Exactly. High ADP means high demand, high respiration rate. Low ADP means low demand, low respiration rate. We can describe this using different states of respiration.
The 5 states. Can you quickly outline those?
Sure. State 1, low everything, low substrate, low ADP, very slow. State 2, got substrate, but low ADP, slow. State 3, the active state, plenty of substrate, plenty of ADP, respiration going as fast as the chain can handle. This is where you measure maximal capacity.
Okay.
State 4,
Yeah.
the resting state, got substrate, maybe oxygen, but very little ADP because ADP levels are high. Respiration slows right down because the proton gradient builds up really high, making it harder to pump more protons. It's like back pressure.
Waiting for demand. And state 5.
State 5. Uh-oh, running out of oxygen. Doesn't matter how much substrate or ADP you have. If there's no final electron acceptor, the whole chain backs up, respiration stops.
So in most of our cells, most of the time, are we hovering around state 4, maybe kicking into state 3 when we get active?
Pretty much. State 4 is the baseline ready state. An increase in ADP flips it into state 3. It's a very elegant feedback loop. And the transporter that brings ADP in and takes ATP out the adenine nucleotide translocase, or ANT, can also influence the rate.
Right. ADP has to get into the matrix.
Yeah.
And you mentioned efficiency. Not all the energy becomes ATP. Some is heat.
Yes, no energy conversion is 100% efficient. The stepwise energy release in the electron transport chain is controlled, but some energy is always lost as heat. This actually helps maintain our body temperature, and it ensures the overall process is strongly exergonic, thermodynamically favorable to proceed.
Okay, tight control, feedback loops. But what happens when things interfere? Let's talk inhibitors and uncouplers.
Right, things that disrupt this beautiful system. Inhibitors block specific steps. Uncouplers break the link between electron transport and ATP synthesis.
Let's start with inhibitors. Where can they hit the chain?
All along it. Complex that can be blocked by things like rotenone, a pesticide, or barbiturates like imobarbital. They stop electrons getting from Fs clusters to CoQ.
Okay, block at the start for NADH. Complex 2.
Malonate is a classic inhibitor. It looks like succinate, the normal substrate, so it binds to the active site of succinate dehydrogenase, part of complex 2, and blocks it competitively.
Complex.
Inhibited by antimycin A or dimercaprol, they mess with the Q-cycle electron flow.
And complex 4, the final step.
Very sensitive. Cyanide CN, carbon monoxide CO, hydrogen sulfide H2S. These bind very tightly to the iron in the heme A3 Cub center, blocking oxygen binding and stopping electron flow completely. Extremely toxic, shuts down respiration fast.
Nasty stuff. Are there inhibitors that target ATP synthesis directly, not the electron chain?
Yes. Oligomycin clogs the proton channel, F0 Lero, of ATP synthesis itself. Protons can't flow back, so ATP synthesis stops, and because of the coupling, electron transport backs up too. Another key one is atractyloside. It blocks that ANT transporter we mentioned, the one that swaps ADP and ATP across the inner membrane. No ADP getting in, no ATP getting out. Shuts down synthesis.
Okay, so inhibitors cause a blockage. How do uncouplers work differently?
Uncouplers create a leak. They make the inner mitochondrial membrane permeable to protons, allowing them to flow back into the matrix without going through ATP synthase.
Bypassing the turbine.
Exactly. So the energy stored in the proton gradient is just dissipated as heat. Electron transport can actually run like crazy, trying to maintain the gradient. Oxygen consumption goes way up, but no ATP is made.
All that energy just lost as heat. Examples.
The classic synthetic one is 2,4,4-dinitrophenol, DNP. It's lipid-soluble, carries protons across the membrane, was once illicitly used as a weight loss drug, extremely dangerous because of the uncontrolled heat production.
I can imagine. Is there a natural version?
There is thermogenin, or UCT1, uncoupling protein 1. It's found in brown fat tissue. It acts as a regulated proton channel. Its job is specifically to generate heat without making ATP, which is vital for keeping newborns warm and for animals during hibernation. It's controlled uncoupling for thermogenesis.
Fascinating. Controlled leak versus uncontrolled disaster. Now, we've talked about the inner membrane being selective. How do all the necessary metabolites, pyruvate, phosphate, ADP, etc., actually get across it?
Through a whole suite of specific transporter proteins embedded in the membrane. They act like specific gates or revolving doors, because the membrane itself blocks charged things like ions and intermediates.
So specialized carriers for specific jobs.
Exactly. Often they work by exchange and antiporter, swapping one molecule for another, like the ADP2P translocase, ANT. Or sometimes a symporter, moving something along with a proton, like the pyruvate transporter, bringing pyruvate in along with H plus Ba.
Can you give a few key examples?
Sure. There's the phosphate transporter, bringing pi in, needed for ATP synthesis, often exchanging for OH or symporting with H plus weight. The dicarboxylate transporter swaps things like malate for phosphate. The tricarboxylate transporter swaps citrate for malate. The iketoglutarate transporter swaps iketoglutarate for malate.
Lots of swapping cycles running.
Absolutely. And critically, the ANT swapping ATP out for ADP in, plus systems for getting fatty acids in, the carnitine shuttle, and transporters for amino acids, and even systems to regulate calcium levels within the mitochondria. It's a busy transport hub.
A highly coordinated traffic system. What about ionophores? You mentioned them briefly.
Ionophores are small molecules, often made by microbes, that can essentially dissolve in the membrane and shuttle ions across, disrupting gradients. Valinomycin, for instance, carries potassium ions. Uncouplers like DNP act as proton ionophores. They're useful tools in the lab, but generally bad news if they get into your cells uncontrolled.
And that transhydrogenase.
Right, proton translocating dehydrogenase. It's another intermembrane enzyme. It uses the energy of protons flowing down their gradient into the matrix to drive the conversion of NADH and NADP+ into NAD+ and NADPH. So it makes NADPH inside the mitochondria, which is needed for biosynthetic reactions and protecting against oxidative stress.
Okay, one more transport issue. NADH made outside the mitochondria, like in glycolysis, can't just cross the inner membrane, right? How does electrons get used?
Correct. NADH itself can't cross. So cells use substrate shuttles. These are sets of enzymes and transporters that effectively pass the reducing equivalents, the electrons, from cytosolic NADH to carriers inside the mitochondrion without the NADH molecule itself crossing.
Clever workaround. What are the main shuttles?
Two main ones, the glycerophosphate shuttle and the malate aspartate shuttle.
How does the glycerophosphate shuttle work?
It's common in brain and white muscle. Cytosolic NADH reduces dihydroxyacetone phosphate, DHAP, to glycerol-3 phosphate. This glycerol-3 phosphate diffuses to the inner membrane, where a different FAD-linked enzyme oxidizes it back to DHAP, passing the electrons directly to FAD, making FADH2. This FADH2 then feeds into the electron transport chain via complex 2 CoQ.
So it effectively turns cytosolic NADH into mitochondrial FADH2.
Exactly, which means you only get about 1.5 ATP per cytosolic NADH using the shuttle because it bypasses complex I proton pumping.
Less efficient. What about the malate aspartate shuttle?
This one's more complex but more efficient, found in heart, liver, kidney. Here, cytosolic NADH reduces oxaloacetate to malate. Malate can be transported into the matrix via the malate ketoglutarate transporter. Inside, mitochondrial malate dehydrogenase oxidizes malate back to oxaloacetate, generating NADH inside the matrix.
So this delivers the electrons as NADH inside.
Yes, which then goes to complex I's, gets the full proton pumping, yielding 2.5 ATP. But you need a way to get the oxaloacetate effectively back out to the cytosol to keep the shuttle running.
How does that happen? Oxaloacetate itself doesn't cross easily.
Right. So inside the mitochondrion, the oxaloacetate is transaminated, swaps an amino group with glutamate to form aspartate and ketoglutarate. Aspartate can be transported out via the glutamate aspartate transporter. Ketoglutarate also moves via its transporter. Once outside, aspartate is transaminated back to oxaloacetate, regenerating it for the shuttle. It's a more elaborate cycle involving transamination and multiple transporters.
More steps, but higher energy yield. Makes sense for tissues with high energy demands like the heart. Okay, one last shuttle system. Creatine phosphate.
Right, the creatine phosphate shuttle isn't about NADH, it's about rapidly moving high-energy phosphate groups. Very important in muscle and brain where energy needs fluctuate rapidly.
How does it function?
ATP is made in the matrix, transported out to the intermembrane space by ANT. There, an enzyme called mitochondrial creatine kinase, CKM, uses that ATP to phosphorylate creatine, making creatine phosphate. Creatine phosphate is small and zips out through the outer membrane into the cytosol.
So it exports energy as creatine phosphate.
Yes. Then, at places in the cytosol where ATP is needed urgently, like near muscle fibers, cytosolic creatine kinase isoforms, CKC, use the creatine phosphate to quickly rephosphorylate ADP back to ATP right on the spot. It's like an energy buffering and transport system.
A rapid local energy delivery service. Brilliant. We've really covered the machinery in depth. Now, connected back to health and disease. What happens when this intricate system breaks down?
Well, as we hinted at, the consequences can be severe, leading to mitochondrial diseases. These can be inherited, caused by mutations in either mitochondrial DNA, which codes for some respiratory chain subunits and tRNAs, or nuclear DNA, which codes for the vast majority of mitochondrial proteins.
And these diseases affect energy production.
Profoundly, often hitting tissues with high energy demands of the hardest: brain, muscle, heart, kidney. Conditions like fatal infantile mitochondrial myopathy or MELAS syndrome, that's mitochondrial encephalopathy, lactic acidosis, and stroke-like episodes are devastating examples, often linked to defects in complex I or IV. You get energy failure, buildup of metabolic intermediates like lactic acid.
And it's not just rare inherited diseases.
No, there's growing evidence linking more subtle mitochondrial dysfunction, maybe reduced efficiency, increased oxidative stress from leaky electrons, to common age-related diseases like Alzheimer's, Parkinson's, type 2 diabetes, even aspects of aging itself. And of course, as we discussed, many poisons and drugs exert their toxicity by directly hitting oxidative phosphorylation. Cyanide is an extreme example.
It really underscores how central healthy mitochondria are.
Absolutely fundamental.
Well, this has been an incredibly thorough journey through the cell's power plant. Let's try to quickly crystallize the main points for everyone.
Okay. We saw mitochondria as double-membraned organelles, with the inner membrane and its cristae being key.
We followed the electron transport chain complexes I through IV, plus CoQ and cytochrome C, passing electrons from NADH and FADH2, ultimately to oxygen.
And crucially, using the energy released to pump protons out of the matrix, creating the proton motive force.
Then ATP synthase, that amazing rotary motor, harnessing the proton flow back into the matrix to generate ATP.
We looked at respiratory control, how ADP levels tightly regulate the whole process to match supply with demand.
And how inhibitors can block specific steps, while uncouplers disconnect electron transport from ATP synthesis, often dangerously.
And finally, the vital roles of membrane transporters and substrate shuttles to move everything needed across that selective inner membrane. It's an incredibly intricate, efficient, and regulated system.
It truly is. So, considering all this complexity, all the interacting parts, the tight controls, here's a final thought for you, our listener, to ponder. What might be the real long-term effects of subtle, maybe chronic disruptions to this system? Think about low-level environmental exposures, maybe dietary factors, or just the accumulated wear and tear of aging affecting mitochondrial efficiency or increasing oxidative stress. What could that mean for overall health, for chronic disease development, for the aging process itself?
That's precisely where a lot of cutting-edge research is focused right now. Understanding those subtle, long-term impacts is a major frontier in biology and medicine. Definitely something to keep exploring.
Indeed. Thank you for joining us on this very deep dive into the engine of life. Until next time, keep questioning, keep exploring.